Jennifer is going to give a talk in her own inimitable fashion, and I think it's going to be accessible to everybody in the audience, which is what we want these talks to be. Go ahead.
Okay, thank you very much for the wonderful introduction, and thank you so much to the Sievers family for being here. It's really an incredible honor to receive this award. And so to tell you a little bit about the context in which this award will affect my research, I will tell you a little bit about how I became a neuroscientist, sort of my formative years. I'll try and tell you embarrassing stories and show you baby photos and the works. Then about how I came to McGill and discovered the Montreal Neurological Institute. I'll give you an introduction to brain science as I was introduced at the Neuro. I will tell you a bit about my thesis work and what I intend to do with the Anne and Richard Sievers Award this year. So, is there a pointer? Or maybe a laser pointer? Okay, so I grew up in a family here. This is me on the bottom, left. And with 4 brothers and 1 sister. My father is a materials scientist, and my mother is a special ed teacher who would help students with handicaps to learn. And when I was an adolescent, she got a degree in therapy, and I'll tell you a little bit about that. So I like to think that there's sort of a genetic and an epigenetic basis for my bias toward brain science. So as a child, my father gave my brother and I a microscope, and we would just spend countless hours collecting all sorts of samples from the plant and animal kingdoms, looking at the exquisite detail that you couldn't see with your naked eye. You know, the cellular components, the building blocks of nature, which are so beautiful, just to look at. My sister Misty was really a world-class gymnast. She was a wonderful, wonderful gymnast. Her focus was on the balance beam. She could do flips and backflips and splits and what have you. And when she was about 17 years old, she started making more mistakes in her routines and having some muscle weakness along one side. And then she started having seizures. So she went to a neurologist to try and figure out what was going on. And due to the low resolution techniques that were available at the time, they weren't able to detect her brain tumor for another 2 years. She was just on anti-epileptics. So after 2 years, they found a tumor in her motor cortex, which was affecting her ability to move. And so they removed it, and they were able to buy her another 10 years of life. So as children, my brother and I built a toy magnetic resonance imaging machine to try and collect data that would help Misty. So in my own adolescence, I became very interested in the humanities, things which humans uniquely are capable of doing, like Mr. Sievers said. Aside from science, poetry, literature, art, music, dance, and theater. So as my mother did her master's degree in psychology, and to help her study for her exams, I would read portions of the Diagnostic Book of Mental Disorders, and I would act out the different syndromes. So without telling her, she would come home to find me displaying the various symptoms of schizophrenia, or being suddenly socially withdrawn and having stereotyped behaviors as in autism. Alzheimer's disease took a long time to act out. At first, it's just subtle changes in memory and mood, and over a course of time, it resulted in complete devastation of memory and an inability to recognize any of my family members. So I came to McGill to do a Bachelor's of Arts, and it was a kind of choose-your-own-adventure major. I studied the humanities, and in my 3rd year, I walked up the hill on University Street to come to the Montreal Neurological Institute and take a course with Michael Petrides on human cognition in the brain, and I encountered this sign. The problem of neurology is to understand man himself. And I thought, this is it. This is what I want to do. This is wonderful. And interestingly, Wilder Penfield, the founder of the Institute and the author of this quote, is the one who discovered, mapped the motor cortex, which I'd been familiar with since I was a child. So what I've learned is that neurons, or brain cells, are the basic units of the brain. They have dendrites, where they receive inputs from all other neurons, and they have an axon, which generates the output. So the inputs are integrated over different morphologies of dendrites, which then makes a decision whether to send a signal through the axon or not. Neurons don't function in isolation, but they integrate into networks of local networks of task-specific cells, which are connected by synapses. And here's one of David Coleman's beautiful images of a synapse, where you can see the axon of one neuron releases neurotransmitter just closely opposed to a dendritic domain that's specialized to receive neurotransmitter. And in this way, through synapses, neurons connect into these functional units capable of processing really complex information. So these local networks of neurons are connected over long distances into long-range networks that are capable of integrating sensory information, producing productive outputs in the form of behaviors, and forming memories. The memories are stored in these connections. So in Alzheimer's disease, what we know is that the proteins, amyloid beta and tau, are expressed in abnormal aggregates. There's a loss of synapses. The neurons atrophy, degenerate, and die. And this results in a gross phenotype of having a brain which is shrunken, a cortex which is shrunken. So my own thesis supervisor, Tim Kennedy, while he was doing his postdoctoral research, was involved with a team who discovered this molecule called netrin-1, which is Sanskrit for one who guides, because it's known to guide axons from neurons which are situated in the dorsal spinal cord here, down toward the ventral midline to cross. And here's the netrin expression here. So what we know, during embryonic development, netrin causes neurons to grow, adhere, and stay alive. Interestingly, though, netrin is still expressed after embryogenesis. So it was my project to look at whether netrin plays a role in the formation of synapses and the formation of circuits in the brain. So here's just one example of some of my data. It's a cortical neuron, which is stained in red for the presynapse, or the axon, and in green for the postsynapse, or the dendritic domain. And everywhere you can see red and green meet are synapses. It's a confocal microscope picture. So what I've done was just to increase netrin, or to block netrin, and to stain for synapses and see whether there was a difference in the number of synapses or the size of synapses. So what you can see here is a quantification of synapse density. Here's the control condition, just with the vehicle alone. And when I add netrin, when I increase netrin function, there's a statistically significant increase in synapse density, whereas when I block netrin, there's a decrease in the synapse density. Correspondingly, and I think you can probably see it in the image, when I look at the area of synapses, increasing netrin function increases the size of synapses, whereas blocking netrin decreases the size of cortical synapses. So as we all know, you can't just have this one piece of data. You have to corroborate it using other pieces, other methods. So working in a laboratory of Dr. David Stellwagen, I sought to do electrophysiology, which is a method whereby using patch pipettes, you can record the synaptic currents. You can listen in on brain cells speaking. So every time you see a little blip here, it's a synaptic event. It's one neuron saying something to another. So I used the same experimental paradigm, increasing or decreasing netrin function. And what you can see is that there are more synaptic currents and that they are larger when I add netrin, and there are fewer synaptic currents when I add the function block, when I block netrin, and they appear to be smaller. So when I quantified this is the inter-event interval, which is inversely related to the frequency. So in other words, adding netrin decreases the amount of time between synaptic events, or makes them happen more often. And when I block netrin, it increases the time between events, so it makes them happen less regularly. And when I look at the amplitude, or the size of the events, increasing netrin increases the amplitude, whereas blocking netrin decreases the amplitude, which corresponds nicely with the immunostaining data. So next, what I did was to, again, add netrin or block netrin following a time course of 15 minutes through 72 hours. And look at the morphology of the neurons. So Here, what you can see is that adding netrin increases the complexity of branching, whereas blocking netrin decreases the complexity of branching. So interestingly, a biochemical interaction has recently been reported between netrin-1 and one of the proteins known to be deregulated in Alzheimer's disease. And it strikes me that actually what I've discovered that netrin does is very much the opposite of what happens in Alzheimer's brain. So I'm interested in this hypothesis that Alzheimer's disease may be related to a loss of netrin function. So what I plan to do this year is to determine if dysfunction in the netrin-1 signaling pathway results in Alzheimer's-like pathology. So if I block netrin in adult neurons, do I see synapse loss, atrophy, degeneration, and death of the neurons? So in summary, I would like to thank Mark and Susie Sievers, the Sievers family, for their incredible generosity and foresight into helping neuroscience research. It will benefit us all, I hope, in the future. And Mark's parents, Anne and Richard Sievers, for raising a son that appreciates the finer things that humans do. I'd like to thank my thesis supervisor, Tim Kennedy. I would like to thank Ed Ruthazer and Dave Stellwagen and Claudio Coelho for their collaborations, as well as all the members of the labs who have been extremely collaborative and very helpful. I'd like to thank my own family and the people I've come to call family here at the Neuro and at McGill. So thank you very much.